Good afternoon, everyone. I'm Jiangtao Gou. I'm a PhD student in statistics at Northwestern. Allow me to assume that everyone here hates disease. Yeah, I think that's the truth. So people suffer from the disease during their lifetime, and probably lives will be terminated by severe diseases. So how should we deal with it? The answer is a fight. All the names on the screens are the big pharmaceutical companies. They spend billions of dollars per year to develop new medicines and new treatments. Some people may think that a new medicine will get into the market directly from a pharmaceutical lab. Then they can purchase it in a pharmacy. The story is not that simple. Why? Because before a new medicine gets into the market, they must be approved by FDA, the Food and Drug Administration. Because we people use medicine, so FDA is very strict and very conservative. Unless you can provide strong evidence to show that your medicine is safe, effective, and better than the traditional treatment, the FDA will not allow the medicine get into the market. So how to persuade FDA? The answer is strong evidence. So all new medicines after they are developed, need to pass the animal test to check their toxicity at the very beginning. Then medical researchers will test the safety on a small number of people, usually between 220 and 80. This is called phase 1. Then they will test the efficacy of this new medicine on a relatively larger amount of patients, usually between 100 and 300. This is called phase 2. And finally, they will confirm the new medicine's safety and monitor the side effect and compare this new medicine with commonly used medicines usually on a large group of people, usually between 1,000 and 3,000. This is called phase 3. So the phase 1, 2, 3 together called a clinical trial. The clinical trial usually will take 10 to 20 years and will cost millions and billions of dollars. And only one of 10,000 new medicines can pass all the tests and finally got the approval from the FDA. So in each phase of the clinical trial, we will use a statistical procedure to test the data. If we can find a statistical procedure with more power, it is possible for us to shorten the clinical trial, save time and money, and reduce the number of participants. So our research is try to develop a new statistical procedure with more power. So let's first introduce the P-value-based statistical test, which is a single hypothesis test. So let's assume that you want to convince FDA that your new medicine is better than the traditional ones. Because FDA is quite conservative, so it will assume that actually there is no difference between these two medicines. And you want to show the strong evidence to them. So what is the strong evidence? So assume under a certain assumption is true, and some very unlikely observation actually occurs. That is a strong evidence to show that your previous assumption actually is not true. So the measurement of the unlikeliness is called p-value. The smaller the p-value is, the more unlikely this observation should occur, and the more strong evidence you will have to show that your previous assumption is not correct. So practically, the FDA will assume that there's no difference between these two medicines. You can convince the FDA only if you have a small p-value, which is smaller than a pre-selected boundary. We call it a significant level. Only in these conditions, the FDA will think that your medicine is better than the traditional ones. But in the real case, a medicine can be used for several diseases. So instead of using single hypothesis test, we need to use multiple comparison test. And we need to adjust the p-value or the significant level. Why? Because if we do not adjust the significant level or p-value, if we test more and more times, it is very likely that we can find some significant result. So let's make an analogy. So here is a garbage can. Okay. So, and I stand this close to it. And I try to throw a paper ball into this garbage can. So if I throw it only once, I think it is very unlikely for me to fail, right? So, yeah. But if I repeatedly throw maybe 1,000 times, I throw 1,000 times, okay. And I've not thrown 1,000 times. Okay. Let me fix this last one. Okay, burn. So I got what it is, if I throw 1,000 times, it is likely that I will fail once or twice. That's why we need to make the adjustment when we do the multiple comparison test. There are some formulas on the screen, but I do not need to read it. Let me explain it. So in the procedure in the clinical trial is called a step-up procedure. Basically, we need to compute a set of critical constant. We labeled as C1 from C1 to Cn as adjustment parameters. Then we need to compare the p-values with the product of these critical constants and the significant level from the largest p-value to the smallest p-value to make the decision. The traditional, the traditional step-up procedure is relatively conservative. So our research developed a new step-up procedure which is less conservative than the traditional one. It means that it